Okay, so here I'm going to go over some common structural motifs in transcription regulators. So if we move down here, the little diagram we have is called the tryptophan repressor. And over here we have the structure of the lambda crow. And right here we've got the lambda repressor fragment. Lambda repressor fragment. And lastly here we have the cap fragment. And over here we just have some DNA, and this is going to kind of give you a size comparison. Like from here to here it's just about 3.5 nanometers. And I know I forgot to label it, but these several proteins are known as helix-turn-helix proteins. Now, originally this motif was found only in bacterial transcription regulators, but there have also been many found in DNA-binding proteins in both eukaryotes and prokaryotes. And they are easily identified by their two alpha helices, the red and the blue, as you can see here. Okay, and moving on down here to the left, on the bottom left we have our leucine zipper protein. Now, the leucine zipper motif was named after the way that the two alpha helices actually interact with each other and the shape that they create when they interact. And you kind of see it's like this X shape, and it's actually in that extended little Y shape that's where the side chains go and contact the major groove of the DNA and interact with it. And over here on the right, we have what's known as the homeodomain proteins. And interestingly enough, homeodomain proteins were discovered shortly after we discovered some of the first genetic regulators in bacteria. And it was through research into the common fruit fly Drosophila that led to the characterization and classification of the homeodomain proteins. And here in the diagram, we actually just get a look at the same molecule from a different perspectives. Like, it's just the same thing, just twisted, basically. And now for the last image that I'm going to go over, down here, it's called the beta sheet DNA recognition proteins. And what's interesting about this motif is that instead of having that alpha helical motif structure that we were seeing before, these rely on a two-stranded beta sheet with amino acid side chains. And they use these beta sheets to interact with the major groove, and that's how they interact with the DNA.